Hello, everyone, and welcome to the second installment of the AGENE webinar series. Today's topic will be process control strategy. My name is Michael Lemke. I'm the Senior Director of Science and Industry Affairs at the Alliance for Regenerative Medicine. I'm going to provide some brief introductory remarks and introduce our speakers. So for those of you who may not be familiar with AGENE, this is a case study that was developed by the Alliance for Regenerative Medicine. The idea was first conceived of at an ARM-CMC workshop in December 2017. Some of ARM's members at the time who had come from the monoclonal antibody industry identified a document called AMAB that had been produced a number of years before that as being one of the things that really helped that industry advance to where it is today and overcoming many of the issues they had that I think bear some similarities to the issues we're currently struggling with in gene therapy manufacture. AGENE again is a case study of AAV manufacture using a triple transfection HEK 293 cell line, employing quality by design principles. The document employed input from over 25 ARM member companies and subject matter experts from those companies. It was made possible by funding from NIMBLE, and it is currently open source, available on ARM's website to be downloaded, the full document at the attached link. These are the chapters you can see. Today's coverage, again, will be on Chapter 7, Control Strategy. For those of you who are coming into the webinar for the first time or didn't see the first one, we're not doing these in exact numerical order, so don't worry if you think you missed something. There will be coverage of the other chapters in future webinars. But again, today's webinar will be on Chapter 7. The educational rollout for AGENE will consist of webinar series for ARM members. These will happen approximately monthly. So we had one in June. This is the second one. We're going to take the month of August off, and then we will resume in September with monthly webinars on other chapters. Culminating early next year in a half-day virtual workshop. Our hope is that AGENE will act as an educational piece and also it will facilitate communication between people who may not be familiar with the principles of quality by design and those who may understand QBD but may not have as much experience in gene therapy manufacture. They're coming from the biologics industry, for example. And we also hope that it will also serve as an educational tool for regulators. With that, I will conclude my opening comments. I would like to introduce you to the presenters today. Irina Sanders, PhD, the Pharmaceutical Sciences Team Lead at Pfizer. Irina has over 10 years of biotech and pharmaceutical experience. She received her PhD in biochemistry from the University of Oklahoma and completed her postdoctoral fellowship in virology at the University of Pennsylvania. She has significant experience in gene therapy development, manufacturing, and testing, as well as leading early stage projects in gene therapy, immunotherapy, and monoclonal antibodies. Also with us today from Pfizer is Althea Mickelwright, the Director, PSQA Business Excellence Lead. She resides in St. Louis, Missouri, and has 25 years of experience in the pharmaceutical industry, and has held positions in a variety of roles in research, manufacturing, and quality assurance, all in the biotech arena. She's spent the last 7 years providing quality leadership for business development, which has included onboarding new gene therapy facilities and developing internal processes and procedures to incorporate this new modality for Pfizer. She has extensive experience moving other biotherapeutic compounds from discovery through product launch. Her education is a BS in chemical engineering from the University of Missouri and an MS in chemical engineering from Washington University in St. Louis. With that, I will turn over to your presenters for the day.
Thank you, Mike, for introduction.
So just 1 second, I'm going to share my screen. And with that, Irina?
Hi, everybody. This is Irina Sanders, and today during this webinar, we will be covering the following agenda topics. We will explain what's control strategy and why it is needed. We will review the elements of control strategy and also discuss how product quality attributes are assessed. We will be talking about material risk assessment, process risk assessment, and also discuss clinical supply chain strategies. At the very end of our webinar, we will provide closing remarks and we'll be able to answer your questions. Next. So what is control strategy? Control strategy is defined by ICH Q10 as following. Control strategy is a planned set of controls derived from current process and process understanding to ensure process performance and product quality. These controls can include parameters and attributes related to product substance and drug product, facility and equipment, in-process controls, finished product specifications, and associated method and frequency of monitoring and controls. When a company develops a control strategy, the company basically wants to ensure the understanding and control of the critical quality attributes to produce a consistent, quality, safe, and efficacious medicine for patients. So in order to have a control strategy, a company would require product understanding and a process understanding. Next. Control strategy in general has 8 elements that fall into 3 major buckets. Bucket number 1, it's a process controls, and they include in-process control of product quality attributes, control of process parameters, and control of process performance. Second bucket, it's a product controls, which include non-routine characterization testing, product controls through quality attribute testing, and product controls through stability testing. And finally, this third bucket, it's a facility and equipment controls, and they include a facility and equipment controls and also controls of materials. Next. So let's review in more detail what types of controls can be done for each element. So here are the first process control bucket, and they involved 3 types of controls. Element 1, which is in process control of product quality attributes. And as far as of types of controls, it includes either direct in-process testing or surrogate testing of product quality attributes and their control limits. Next type of control in this category is process validation. Element 2, no, can you go back? Element 2, which involves controls of process parameters. And types of controls, it's either control implicit in the design or manufacturing process or unit operation, manufacturing process controls, controls of limits and acceptance ranges, manufacturing whole type control limits, and manufacturing process development and history. And element 3, which is control of process performance, involves in-process test of process performance and their control limits. Next. So the second bucket, it's product controls, also has 3 elements. Element 4, which is non-routine characterization testing, has the following types of controls: elucidation of structure and other characteristics, non-routine testing for characterization and demonstration of product comparability, and also characterization involved in reference standard and reference standard qualification. Element 5, which is product controls through quality attribute testing, can involve either routine release testing and acceptance criteria in product specification, justifications of the specifications, analytical procedure and its validation, product quality attribute control demonstrated through process validation. And then element 6, which is a product control through stability testing, involves the following types of control: routine stability testing and acceptance criteria, instability protocols, and also stability data and their conclusion. Next. The final bucket, which is a facility and equipment controls, has only 2 elements. Element 7, which is a control of materials, and it has a variety of different types of control. For example, specification for raw materials, manufacture and testing of cell banks and cell banks controls. It also involves characteristics of incoming materials such as raw materials, starting materials, intermediates, and packaging materials. Everything that potentially can impact product quality attributes in the acceptance ranges. And the final types of control for this category, it's compatibility with container closure system and container closure controls. And final elements, which is a facility, can you come back? Facility and equipment controls involve CGMP and pharmaceutical quality systems for the manufacturing facilities, and also environmental and equipment controls directly impacting product quality attributes. Next slide. So what are product quality attributes? Quality attributes, or QAs, describe the physical, chemical, biological, and microbiological properties of the product. And these quality attributes may change during production or storage or administration. And QAs might have a positive, negative, or no effect on product quality and efficacy. So for AAV-based gene therapies, we identified a variety of different quality attributes. I listed here a subset of them, but there are more than this list. And as you see, it can be as a particle content, aggregation, sterility, bioburden, potency, residual DNA, residual plasmid, and much more. Next slide. So usually, in relation to quality attributes, we need to assess them. And the quality of assessment, so the goal of this assessment is really identify and control attributes that are critical to product quality, safety, and efficacy. And a key word is critical. So by determining the criticality of quality attributes, we basically support many aspects of product development. And assessment of criticality occurs during product development and usually based either on prior knowledge or publicly available information. Quality attributes are scored based on two parameters, which is a potential impact or severity of the attribute, and also on uncertainty associated with this impact. And a quality attribute will be considered critical quality attribute when evaluating in these two categories. The score that we get will exceed a predefined criticality threshold, and that accounts for the combination of severity and uncertainty scores. Also, FDA defines the critical quality attributes as physical, chemical, biological, and microbiological property or characteristics that should be within the appropriate limit or range to ensure the desired product quality. So any QAs that are measured by compendial assays are automatically considered as critical quality attributes. Next slide. So how do we usually in practice assess product quality attributes? We actually, for both severity and uncertainty, we have a scoring system. On the left top side, you see a scoring system for severity of CQAs, and it can be broken down into 1, 5, 7, 10, with 1 is negligible, meaning no harm to the patient, and with 10 is a major severity, meaning with irreversible effect on patients or potential serious impact to a patient. The same scoring system exists for uncertainty. Again, it can range from 2 with a prior knowledge and well-established understanding to 2 to 10, which means low confidence and information. So on the left bottom, you can see how basically we create a matrix and we can predefine which of these quality attributes will be potential CQAs, and they will be shown in red. They basically need to exceed certain thresholds that are predetermined by a user. Next slide. Also important, can you come back? Also important to point that in AAV gene therapy, uncertainty is so high that potentially everything is a critical quality attribute. It's just very, very specific for this particular modality. Next. So let's continue with how we can actually control and assess quality attributes. It can be done, for example, in process control of product quality attributes. In process control, no, can you come back? No, just one click forward, yeah. So when you have in process controls, you are able to have kind of a live control of your quality attributes. Let's say you have a sample drug substance and sample drug product process. And just to provide you a couple of examples of what in process controls might look like. I'll see a next click. For example, this for drug substance manufacturing, for unprocessed bulk, it can be bioburden, mycoplasma, and advantageous agents. For harvest, that can be qPCR, bioburden, endotoxin. For affinity purification, qPCR, bioburden, endotoxin. For example, for ion exchange chromatography, bioburden, endotoxin, and the same is for UFDF. The same is for drug product. Next click. For example, for pre-sterile filtration, it can be bioburden measurements. For sterile filtration, it can be pre and post-use filter integrity testing. And for sterile filling, it can be fill weight check. Next slide. Next type of control is a control of process parameters. So in AAV manufacturing, unit operations are very similar to any biologics. We have upstream unit operations that focus on production of recombinant viral particles. We have downstream operations that are consisting of purification and polishing steps, and finally formulation and fill. Unfortunately for AAV, the relationship between process parameters and CQAs are very complex and not fully understood due to the kind of a new nature of the modality. So in order to elucidate this relationship, companies usually conduct process development and small-scale qualification. Also, for control of process parameters for gene therapy, any company can use pretty much any well-known and established process control strategies, for example, from any new modalities. For gene therapy, one step is the most challenging for process control is transfection, because in triple transfection, the efficacy or efficiency of the transfection is very dependent on multiple factors and process parameters. Also, what's important for AAV-based gene therapy is that critical doses are very high, and usually productivity are relatively low. In this case, in order to, for example, reduce material for release test instability and retains, multiple DSs can be pulled into multiple sublots can be pulled in this one drug substance lot prior to convert it into the drug product. Next slide. Next type of control is a control of process performance. So when we control a process performance, meaning its robustness and consistency, that can be established through defined batch instruction in process monitoring or in process testing. And for example, here we give a couple of examples, for example, for cell culture and for drug product filtration and filling process performance. For example, for cell culture, it can be incubation, CO2 concentration and temperature, media equilibration temperature and duration, cell count viability. And for example, for drug product sterile filtration, it can be drug substance thaw temperature, filtration pressure and flow rate, mixing temperature and time. Next slide. Next type of control is non-routine product characterization testing. Well, some attributes are actually better characterized via heightened characterization testing instead of routine GMP release testing. So usually these methods are not used for routine testing, or some of them are not actually even GMP compatible. However, they do provide very important supportive information that helps defining additional elements of control strategy. And here on the right, we show some of the example of non-routine characterization testing and why we potentially use certain tests. For example, if the quality attribute is capsid size, and we want to use analytical testing, which is DLS, can you come back?
Oh, sorry.
So the rationale for it that is properly formed capsids are expected to have consistent size, which can be measured by DLS, and this is not a GMP compatible test. For primary capsid sequence, we can use peptide mapping, which is allows us to evaluate primary capsid structure, which ensures capsid identity and enables characterization of post-translational modifications. And non-routine product characterization testing also helps to link all comparability studies in a program, from toxicology studies to commercialization. Next slide. Althea?
Yep, thank you, Irina. Sorry about skipping your slides. Not very skilled at this. So now we're moving on to talk about elements 5 and 6, which we've kind of combined together here just for simplicity's sake. And so we've combined the product control through GMP release testing as well as the stability. And so what we're showing you here is just one example of what a potential control strategy could be looking like for any AAV gene therapy program. However, every project should be independently evaluated by incorporating justification of the specification to define the routine GMP release testing and the acceptance criteria in a phase-dependent manner, because it does really matter whether you're in phase 1, phase 2, phase 3, or trying to do your final launch. So normally, as you would progress through the product life cycle, the analytical methods should initially be qualified and later fully ICH validated as the program enters into the process validation stage. That typically would occur around phase 3. And your stability protocols will include relevant stability indicating assays, and then your use period for the drug substance and the drug product will be periodically updated throughout the life cycle as you gain more data. But I would be amiss not to mention the impact of the global pandemic has had on acceleration of program timelines. And I'm sure there's a lot of you out there that have been involved in many of the programs. You know, just as a quick recap, at the end of January 2021, you know, the US government-sponsored program, Operation Warp Speed, provided funding for 5 of the 16 SARS-CoV vaccine candidates that were in phase 3 trials around the world. This also included 2 non-replicating AAV vaccines. We had the AstraZeneca Oxford chimpanzee adenovirus, and we also had J&J with their adenovirus type 26 vaccine. And it's obvious to all of us that regulatory approval for emergency use was gained in record times under the OWS program. And, you know, the acceleration causes many activities to be performed in parallel. And what that has done, I think, is added a really heightened emphasis on developing a robust control strategy. So I'm not going to go into the details of this slide. It's in the chapter, but you can see here it's just kind of like a, you know, one proposal. All of this is up for negotiation with regulators. And so I would just use it as, you know, a guideline, if you will. So going on then, moving from product control into control of materials. I think in the gene therapy space, this is one of the areas where there's been a lot more focus. And so we really wanted to highlight that in our chapter to, help people understand some of the things that were the same, but also some of the things that were really different. And so, in the starting, we kind of put things in three buckets: starting materials, ancillary materials, and excipients. And, starting materials are the starting point at GMP process manufacturing, as I'm sure you're all aware. The thing with gene therapy is that the starting population of cells or plasmids are considered the starting material for gene therapy products typically. But for products consisting of viral vectors, the starting materials could actually be the components from which the viral vector is obtained. So that could be the master virus seed, or the plasmids to transfect the packaging cells, or the master cell bank of the packaging cell line. And ancillary materials, if you guys have worked with packaging and labeling, they've had a definition for many years of what ancillary materials is. But within gene therapy, it's taken on a different definition. And so the defining property of ancillary materials really is that they're not intended to be part of the final product. So they're definitely not a starting material. They're definitely not an excipient, but they do, they're materials that are used in processing and purification aids or agents, and they do exert some effect on the therapeutic substance. So the effect may be a physical or a chemical, biochemical change to the active ingredient, or it could even be an exchange of genetic information that happens, but doesn't necessarily show up in the end product. And so the final bucket there is the excipients, which hopefully you're well aware of these, but the thing about the excipients, there really is no difference in the expectation of what's required for excipients and gene therapy products versus any other biological, biotherapeutic product. So I'm not really going to spend a lot of time on that piece. I'd really like to focus on the ancillary materials because I think that is what is different with gene therapy. Irregardless, for all three material types, you still need to define a formal rationale of what their use and qualification strategy is going to be in a risk assessment, along with whatever the controls and mitigations that you're going to have to employ throughout the life cycle to ensure the quality, safety, and efficacy and traceability, which is a big thing, the traceability of the materials. So with that, I'd like to go into the focus on the ancillary materials. And so there's a lot of expectations, which you can see in our chapter in the references, that can be found in USP 29 under 1043 or 1046. And then we also have the EU guidelines referenced as well. And really, the control strategy can be broken down into 4 categories that kind of go from tier 1 to tier 4, with tier 1 being your lowest risk, your most highly qualified materials. And then, as you go up the tree, you will see that the tier 4 ones require extensive qualification prior to use in manufacturing, especially if they're not produced in compliance with relevant GMPs. And phase-appropriate discussions really need to play in here because, what you need at phase 1 for these tier 4 materials is certainly a lot less than what you're going to need when you're trying to file a BLA or an NDA or whatever, or BLA or the European equivalent. So sometimes a picture is worth 1,000 words. And so we didn't include this in the chapter, but this is kind of a decision tree that we worked out internally to show you kind of like what it could look like to kind of go through and decide whether or not you are at high risk, low risk, medium risk. And so there's some key takeaways here. And the first thing I would point you to is that even if this material is commercially available and there's some material grade assigned to it, it could still end up being high risk, because sometimes the suppliers do not want to commit to what they're making. They're just providing it to you, and it's up to the sponsor to decide how it's being used in their process. And so the commercially available materials sounds great because you think, oh, okay, no problem. But in fact, you really have to go through a decision tree to understand what do we know about that material and how well controlled that material is. And it could put you into a tier 4 high risk. The third thing is, regardless of the material, all of these materials need to be risk assessed from the point of view of how they exert their effect on the therapeutic substance. So this may be a physical or chemical, biochemical change to the active ingredient, or it could be a change to the genetic information. Again, ancillary materials are not going to be found in the final product. So this is why they become so important, because they play a critical role in the process. So some additional considerations with the ancillary materials is really, I know we focus a lot on spec development for drug substance, drug product, even our cell banks, virus banks. But with the ancillary materials, you really have to have your analytical group really focused on making sure that they understand what is the testing strategy and control strategy for the starting materials, the ancillary materials, and the excipients. And, there are some instances that we've seen where we have had some novel excipients being introduced, and those tend to take quite a bit of work. If you guys have worked with making sure you've got your GRAS statements and making sure that there's toxicology data and things like that, there can be a tremendous amount of work on your analytical team to make sure that they have a robust control strategy. Another portion of this is talking about just the retain strategy and the stability programs, because in early phase, you have limited information, you have limited supply, you have, things are really unsure, but you need to have an eye to the future, thinking about what are you going to need when you're successful, right? So when you have to prove comparability, when you have to prove that, what it was yesterday is the same as it is today and the next day, you're going to need some of those retains that we wouldn't normally keep, and there really is no regulatory requirement for you to keep those retains at this point. But you need to be thinking about that, because it's going to come down to, can you show that whole comparability scheme all the way through your whole program? And so there are some critical materials that are in your in-process space that you're going to have to think about. Another thing is, and this was a learning for me going through this, a supplier will give you a use period and you think, oh, okay, that use period is good. Well, they don't necessarily provide you with a use period that is appropriate for your use of that material. And so it really comes back to the sponsor to make sure that the information that you have been provided from the supplier, but also the information works with your program. And so, there could be some additional work on your analytical folks to make sure that they do agree with that use period that's been supplied, if it has been supplied, and in some cases they won't supply it. So then the onus is on the sponsor to, generate that data set. So that's something else to think about with this. You really have to be forward thinking. And with that, I'm going to turn it back to you, Irina, and we'll go to process risk assessment.
Okay. So in order to develop a manufacturing process that is consistent and reproducible, in order basically to deliver the medication to the patient, any company will need to develop an approach to understand the relationship between process input or process parameters and the output attributes. So in order to do this, we conduct multiple systemic risk assessments, and they are done throughout the development lifecycle to identify process steps, material attributes, equipment design, or operational parameters that can be affecting drug substance and drug product quality attributes. Here in this diagram, we show 3 major risk assessments that should be conducted for these purposes. However, additional risk assessment can be done throughout the program life cycle. So in order to do a risk assessment, companies can use different tools. Later on, I will be reviewing 2 of these tools, which is cause and effect matrix and FMEA table. But before we go to the explanation of tools, let's talk about what exactly risk assessment strategy could be. So in a risk assessment 1, usually risk assessment 1 is done prior to or in parallel with the decision to start late-stage development and initiation of moving the project to a commercial manufacturing scale or site. So what basically is done here, we're trying to understand the impact of process parameters on quality attributes based on product knowledge through the cause and effect matrix scoring. And the outcome of this risk assessment is basically we're trying to identify and prioritize a list of lab studies and experimental strategies. So the risk assessment 1 is used to conduct risk assessment 2. During risk assessment 2, companies revise their cause and effect scoring matrix based on experimental output. And they also conduct FMEA risk assessment to understand process parameters effect on critical quality attributes. And as a result of risk assessment 2, usually the list of CPPs and non-CPPs with a potential effect on CQAs. There is companies develop a process performance qualification or PPQ control strategy and define PPQ criteria. And the risk assessment 2 results are usually used for risk assessment 3. Usually risk assessment 3 is done following PPQ and prior to regulatory submission. And during risk assessment 3, again, cause and effect FMEA matrices are revised to understand process parameters effect on CQAs based on the PPQ and other batch experiences. And the outcome of risk assessment 3 is a final list of CPPs and non-CPPs with a potential effect on CQAs and basically a commercial control strategy. Next. So as I already mentioned, for risk assessment, companies use a variety of different tools. Here, I'm going to review 2 tools. It's a cause and effect tables and FMEA table. So what you see here on the screen is an extract from cause and effect risk assessment for AAV affinity chromatography process. And usually, what is usually done in this cause and effect matrix is you basically try to represent all aspects of certain process that can potentially have influence on the critical product quality. And so usually in a matrix like this, you assess the relationship between process parameters or process input and process output or attributes. And these attributes can be either quality or attributes or process performance attributes. So as you see, for example, for process parameters, there is a scoring table. And again, the scoring can be either numerical or it can be low, medium, high. Here I'm showing a numerical scoring system that can be ranging 4, 1, 4, 6, 9, 10. You know, each and every company can actually have their own numerical scores, with 1 having, for example, there is no known relationship, and 10, it's a strong relationship. And a very similar scoring system can be developed for attributes. It can be selected 1, 5, 7, 10, with 1 having no impact on product safety, with 10 having a direct impact on product safety. Then basically, it's usually a team exercise. All team members that are involved in you know, process development or manufacturing, quality representative can be contributing to cause and effect tables. You kind of, in a sense, plot your parameters versus attributes and do the scoring. The numbers that are shown here are not good or bad. They only show the strengths of relationship. So when you do the scoring, at the very end, you kind of collect a total, and this total actually allows to prioritize you know, your studies. So when you do prioritization, you know, this output of this is now going into your design of experiment studies, which will help you to identify, you know, which additional parameters have to be evaluated further. Next. So another tool for a risk assessment is a FMEA tables. And for the FMEA tables, what we're trying to understand is to identify any inadequacy in a process, right? In a process of developing the project. And FMEA is evaluated in terms of degree of severity of consequences of these inadequacies, the respective chance of occurrence, and their detectability. And again, for severity, occurrence, and detection, any company can develop their own scoring system. Here we are showing scores from 1 to 9, with 1, for example, no impact on process performance in severity, never observed in occurrence, and excursion can be very obvious for detection. And it can go to 9, which 9 represents in severity a failure to meet the S&DP specifications. In occurrence, it's very frequent occurrence, and detection, no way to detect excursion, or it's not trackable, or is not alarmed. Next. And Althea, I'm giving it back to you.
Yes, thank you, Irina. So, you know, one of the other things that we wanted to include in this webinar was just really as an extension of element 8, which was really focused on the facility equipment controls, process controls. We also wanted to include some information on some of the challenges around the clinical supply chain considerations that really affect gene therapy products. And I'm sure, as all of you know, recognizing that, you know, you really have small batch sizes, you have limited quantities of drug product available, you have to come up with a material use optimization strategy that can be used to facilitate getting the clinical distribution out to the patients. And so some of the challenges that we found really centered around the drug product manufacturing process. So in our world, we would separate drug product manufacturing was one group, and then the next group was pack and label. And so there was a clear handoff in our legacy operations with just typical biotherapeutic programs. And so what we found with the gene therapy space is that we actually had to move primary labeling up into actually move it into the drug product manufacturing space, and also the inspection portion, which was not typical. So that was one of the big changes that we had to make. But you know, it was really driven from the fact that you know, the drug product had to be frozen as quickly as possible, usually at -70 degrees. So you can't really apply labels once you freeze products. So that was one of the challenges that our global clinical supply group had to work through and work with our manufacturing folks. And so we ended up incorporating, and with regulatory, incorporating a revised approach where we had minimized primary labels that were able to be put on each of the vials, which you know are very small, of the drug product. And then so they would, like, we would do the drug product, we would do our visual inspection, and then we would apply the label and then freeze them. So it took a lot of conversations with regulators because they wanted so much information on that primary label that it just wasn't possible. Because typically on your primary, you still need your protocol number and other things. And so we were able to work through that with them. And this is kind of like a pictorial of what our global clinical supply group came through. But we integrated the drug product manufacturing with a primary label at the site. Then the vials were inspected, then they were frozen down. And what that minimized primary label that we were able to work through regulatory with, it allowed us to ship the semi-finished goods to an inventory spot without being allocated to any particular protocol or country. So that's the first time we've ever had to do that. So I don't know if it's different with any of the other, companies out there, but that was a big change for how we did our operations. And the other thing is only once a subject is identified and gets enrolled at any given site within a protocol does that shipment trigger the next step, which is working with our contract pack and label sites to create that secondary packaging and the kit that's going to go specifically for that patient with that right dose. And so it's very patient-centric, which is, I think, probably the first, at least in our company, it's the first time we've had to do patient-centric work. But it's been working out really, really well. And, some of the additional support that has to be supplied to support the clinical trials is around the dosing and administration instructions, because, of course, we have evolving dose ranges, and, we have to make sure that we've got the right vial thaw process so that we don't compromise the materials. Again, analytical has played a heavy weight here to help us with this and make sure that we've got like instructions on what's the shelf life that can be held at the clinic and what are the storage conditions at the clinical site, and really just documenting what those are for the clinicians to make sure that we're getting the right dose to the right patient at the right time. And so with that, I'm going to turn it back to Irina for some closing thoughts. Thank you, everyone.
Next slide. So, and in the conclusion of this webinar, we would like you to, kind of get the major points that we were trying to educate you today. Point number one, that a critical quality attributes for AAV are still, it's an evolving process. There is a lot of uncertainty, and risk assessment and carefully designed studies are extremely important to advance our knowledge. Another point is a formal risk assessment is expected for ancillary materials. A control strategy needs to evolve with the process and understanding. It's not just a release test with specification. And the distribution to the clinic requires innovative supply chain solutions. Well, with this, thank you so much for your attention. We would like to acknowledge Pfizer team members who participated in creating the chapter. Paul McCormick, Dave Sarelli, Megan Horn, Phoebe Boulders, Agnieszka Bandula, Janelle Lavoie, Janet McEnroy, Arun Alphonse Ignatius, Patrick Forcola, and Joanne Parker. Thank you so much for your attention.
Thank you both, both for your contributions to this chapter and that fantastic overview you just provided of the chapter. So we have quite a few questions in Q&A. I'll try to get to as many of them as possible. I'm going to jump right in the order that they were asked, if that's okay. So first question is, and to put this in context, I believe this came up, Irina, during the first part of your presentation on a slide where you were talking about the effect of uncertainty on CQAs and how it's common to have high uncertainty in the context of gene therapy. And the question is, wouldn't you rather have CQAs where you have a lot of information about them so you can better understand the attributes for release specs?
Well, we might not have a lot of information about CQAs. It's a new modality, so we need to. It's a process. So you have to define CQAs relatively early in the process. I mean, obviously everybody would like to have more time, but it might not be.
Right.
It would be feasible.
Is it correct to say that the process control strategy is a living document or a living strategy, so to speak?
Exactly, it's a living strategy that is that will be evolving with the modality itself.
Right. So for the questioner, we'll get there to where I think you're envisioning us being, but it's going to be a process and a journey. So I'll move on to the next question. Traditionally, which department is the owner of the control strategy? Is it process development or QA or another department?
It might be having multiple owners for the control strategy. It might be simply having, it might be simply a different team that has technical folks and QA folks as a separate team, not necessarily a department. That can be a control strategy group that owns it.
Got it. Okay. Next question is about pooling sublots in order to get a single drug substance lot. And the question is, do you establish acceptance criteria, or have you established acceptance criteria for sublots prior to pooling into a single drug substance lot for filling operations?
Yes, you can, because you obviously would like to have a relatively homogeneous number of sublots that will be going in your final DP.
So maybe if I could chime in there, because this is a very debatable topic about what you need to test. But yes, so you can define what the critical quality attributes are for the sublots prior to pooling. But yes, you do need to have a justified approach that explains what the criteria are, and then use that to justify your sublotting. And it does also require some discussion with regulatory agencies. Not every, I mean, FDA has a different kind of point of view sometimes than Europe. So, I mean, you do need to have those discussions prior to making those decisions.
Okay, thank you. Next question, and I'll add my own addendum to this question. What is the best stage to perform non-routine product characterization testing? And maybe if you could cite some reasons for conducting non-routine product characterization testing.
Well, the non-routine characterization testing is really to expand your understanding of the product. Again, some of the testing that might giving you this extended information are not, you cannot do them GMP. They cannot be even conducted GMP, but it gives you information to expand your understanding of your product. And what was the first question?
At what stage and development is it done, or is it done in multiple stages?
You can start doing hidden characterization even at early stages of your program.
Okay, thank you. So a comment and a question that actually comes from one of your former colleagues at Pfizer, who is a contributor. He says, congratulations to you and the team. And this is kind of an open-ended question. In going through this and doing this work, does it tell you that AAV manufacture has more in common with traditional biomanufacture?
Can I take that one, Irina? So I would say that there are a lot of commonalities. There absolutely are. I think what we tried to elucidate with the control strategy chapter in the book is there is a heightened expectation around not just the ancillary materials, but really having a formal risk assessment that shows that you understand for every material that enters that process, what the impact is on the final product and how it is controlled, and that you've minimized any end product impact with those materials, which I think is kind of more of a heightened expectation than what I've seen in any of the other biotherapeutic programs. And I've worked on mammalian, microbial, vaccine. I mean, I worked on, messenger. I worked on a lot. So, for me, I think in gene therapy space, there is a lot of people, like the regulators, really want to understand that as a company, as the sponsor, that what's going on in your process. And so it takes process is the product to a whole new level.
Got it. So there's a question. I'm going to keep going with the questions, and we have some poll questions for the audience at the end. If we have time, we'll get to them. But I want to make sure we answer as many questions from the audience as possible. The question is, would you provide your thoughts on assembling a summary control strategy document, including a brief summary for each of the 8 elements that you described? And the questioner says they see value in the all-in-one summary for communication, education, and supporting module 3. So if I'm understanding the question, is the value in assembling a summary control strategy document for a BLA as part of a BLA submission?
Yeah, I think we would have to consult with our legal department to do that.
Okay. So perhaps we can circle back on that one, but I'll move on for now. There's a question about the math on the risk assessment. For the person that asked that question, I believe that's explained in the chapter, but we'll circle back on that as well, rather than trying to do the math live here, if that's okay. And another question is about the labeling information. If you're able to answer this, how aligned are EMA and FDA with the approach you presented for labeling? Was it possible to use one label for both regions?
It was.
Okay, that's an easy one. Good.
I mean, it wasn't easy. It wasn't easy. We did finally get there because the EU, they really want to have the expiration on the label, and that's not always possible, especially when you don't know where it's going to. So yeah, that was one of the big challenges, was really around the expiration dates.
Got it. Another question, and they keep coming in. We'll get to as many as possible. Do you have any examples of unexpected or unusual quality attributes, specifically for starting and ancillary materials, and how are they measured?
I can't think of one off the top of my. Rena, do you have an example?
No, I'm trying to think about it.
We could circle back on that one too, Mike.
Yeah, maybe we could circle back on that one.
We might have to consult with some of our experts too.
Okay. Next question: Does the Vial primary label also have patient-centric information, or is it only on the secondary label?
Only on secondary.
Got it. And the second part of the question, have there been any challenges with not having all of the required information on the primary label at the time of drug product manufacturing? If you're able to answer that.
Absolutely. Every program has to have a conversation with the regulators that they're trying to get into that country. It does require regulatory endorsement to make any, minimalization of that label. So yeah, you have to have those conversations.
Okay, so I guess one takeaway is it sounds like there's a lot of work and conversations with the regulators around labeling in general. Okay, so I think we're actually at the hour. So there are a number of additional questions. We will save these and try to circle back to the questioners on them. I think maybe Jancy, if we could just do one polling question, if we could pop that up real quick for in the last 30 seconds. So if we just want to answer the first question, since we're limited on time, which of the following are key elements of the AG process control strategy? A, in-process control of product quality attributes, B, product control through quality attribute testing, or C, non-routine product characterization testing, or some combination of these as described in D and E. You could just answer that. Actually, if you can see the second two questions, if you want to try to answer those as well, feel free to go ahead, and I'll run through the replies, the responses pretty quick in about 30 seconds.
Can everybody see that on my screen, Mike? Maybe that's what happened last time.
Yeah, you should see the polling questions.
Yeah.
Now.
Do you see them on my screen?
Yeah, Jancy, are you getting responses?
Yep, they're coming in.
Yeah, maybe just give it like 5 or 10 more seconds and go ahead and pop them up. All right, so the majority got the first question right. Well done. So it was all of these three elements are part of the aging process control strategy. When is the third risk analysis performed? Nobody fell for the trick answers. 94% picked the correct answer after PPQ, but prior to the regulatory submission. And hopefully everyone got this right. A few of you didn't. So you'll have to watch the webinar again, but true or false, ancillary materials may be considered to be low risks since they are not intended to be part of the final product. That is false. And 91% were correct on that. So most of you, it looks like, paid attention and hopefully got a lot out of this. Thank you all again, and we will circle back on the questions that we didn't get to. And thank you again, Althea and Irina. Take care.
Thank you so much.
Thanks, everyone. Really appreciate you.
Bye.